Hello everybody and welcome back to video 2. This is going to look at the structure of muscle fibers and of course the mechanism by which muscles contract. So if you're a sports science student, you've probably already done quite a lot on this and you'll probably actually know quite a bit more about it than I do. So please bear with it and maybe use this as a refresher for the course. Okay, so what we can see here in this image is a muscle fiber, all right? The individual units of this muscle fiber are called myofibrils, okay, which you can see sort of running down here. They have something around them called a sarcolemma. Now, the sarcolemma is like a cell membrane, okay, and this is why actually muscle fibers are quite different to other types of cells, because the fibers have one cell membrane around them, but within each of those membranes or in each sarcolemma, there is lots and lots of nuclei. So they're not split into individual cells like most cells are split. Okay, so this is one of the cells that kind of breaks that apart, that idea of the cell theory. Okay, and is actually contrasting to the cell theory. So important to remember that the muscles we have are used to move the body and are attached to bones, and so we call them skeletal muscles. If we look using a microscope, we can see stripes. Okay, now you can see these stripes on this image here. Okay, and so sometimes we call this type of muscle striated muscle. The other two types being smooth muscle fibers and cardiac muscle. Striated muscle, which is what we're going to be focusing on today, is made up of these bundles that we can see. And each of the bundle is made up of lots of fibers called myofibrils. So we've got this single cell membrane around it called the sarcolemma, many nuclei within inside it, and each of these fibers is much, much longer than any typical cell you might find in the body. And this is because embryonic muscle cells will fuse together to form these muscle fibers. Okay, so this happens very, very early. Within inside it, you can see this thing called the sarcoplasmic reticulum. Okay, this is a modified version of the ER, or the endoplasmic reticulum, which we find in most other cells. Remember, ER are made for protein, we use them for protein synthesis, the sites of protein synthesis and protein modification. Okay, but when they're modified in muscle tissue, basically they wrap around the myofibrils, so around these individual fibers that we see. And when these actually convey an electronic signal to the myofibrils to contract, and so it allows all the myofibrils to contract and relax simultaneously. These sarcoplasmic reticulum, they store calcium, and there are also large numbers of mitochondria around which are supplying energy which is needed for the contractions. Okay, the idea of what the calcium is for and what the ATP or the mitochondrial ATP is used for, we will come back to very, very shortly. Okay. Again, some people have already done this when they've looked at sliding filament theory in sports, but again, it does no harm to kind of rethink it. All right. Okay, moving on. So here's a close-up of one of those muscle fibers. What you can see is that you've got a light band which has a Z line running through the center. Okay. Interspersed between those light bands and Z lines, okay, is a dark band or a dark band area. So this combination of this dark band area and this light band area, okay, is what we call one sarcomere. Okay, so again, important features are we have alternating light and dark bands. Okay, the whole thing is one myofibril. We've got a Z line in the center of the light band. Okay, followed by a dark band, followed by another light band, and another Z line. And the sarcoma runs from one Z line to the next. Okay, now that's not going to mean a lot to you at the moment, simply because you don't really know what's involved or what's in these parallel lines, and that's what we're going to come on to in our next slide. So in this electron micrograph, okay, this is what you can see here. You can see quite clearly that it shows a longitudinal section through a myofibril, and you can see a number of repeating units that alternate between light and dark. So you've got light part here, a Z line, another light part, dark, going into light, and another Z line running down the center of there. So there, from here to this point here, this is our sarcomere, okay? And basically, what this sarcomere is made up of is a protein, a thin actin protein, and a thick myosin protein, okay? In our next video, or video 3, we're going to figure out the interactions between those, okay? And that's what we're going to have a look at in our 3rd video. So that's it for this 2nd video. Keep watching to see the 3rd one.